Hi guys, welcome to Cosmic Solutions, Dr. Priyanka's Biology, where we are going to discuss everything from biology to biotechnology. So in our previous sessions, we had started with cell cycle and cell division. So where we had discussed about mitosis, the phases of mitosis. Then we had started with meiosis. We had seen the first subcategory or the first subdivision of meiosis, that is meiosis 1. So we had seen whatever happens in your meiosis 1, why the prophase 1 is so complicated, what happens in different phases of Prophase 1. So all these things we had seen. So now in today's session, we are going to start with the second part of meiosis, that is your meiosis 2. So if we could quickly recall what had happened in your meiosis 1, okay. So basically your meiosis, okay, it is for your cells which will undergo reproduction. Basically it is for your reproductive cells, that is the egg or the sperm cell. Okay, so the beauty of your meiosis is that, okay, there will be genetic variation. Okay, so important thing is there is something called as crossing over, which was not there in case of your mitosis. Then there is something called as, formation of chiasmata. So all these things were there. There is something called as homologous chromosomes. So all these things we had seen in case of your meiosis 1. Okay. Then particularly we had discussed prophase 1 in detail because major things are happening in your prophase 1. So from your prophase 1 to your metaphase 1, anaphase 1, telophase 1, we saw there was much similarity to that of your phases of your mitosis, but we had been talking about the homologous chromosomes. Okay, so today's part we will focus on meiosis 2, which is much simpler as compared to your meiosis 1. So we had discussed about something called as your interkinesis, a phase between two successive meiotic divisions, that is your meiosis 1 and meiosis 2. Okay, the short phase called as the interkinesis. Now let's see what happens in case of your meiosis 1. Sorry, meiosis 2. From this picture, okay, we can see that the two cells which were the result or the end result of your meiosis 2, two haploid cells, okay. From a diploid cell, we had got a product of two haploid cells. Now, these two haploid cells, they are going to simultaneously undergo meiosis 2, okay. So, let's see what happens in your meiosis 2. Okay. Now, if you can recall, we had seen that there will be S phase or the cell is undergoing, okay, it will undergo DNA replication only once. That will happen before the meiosis 1, the interphase of your meiosis 1. Before meiosis 1, okay, it will undergo the replication. Okay. Now, cells move from meiosis 1 to meiosis 2 without copying their DNA. So whatever is there in your meiosis 1, same thing is going to enter your meiosis 2. Okay, because we had been talking about the interphase before meiosis. But meiosis 1 and meiosis 2, it's a continuous process. So the sequence is interphase, meiosis 1, meiosis 2. So important thing or important highlight is that this transition is without copying or without undergoing the S phase. Okay. Now meiosis 2 is shorter and simpler as compared to your meiosis 1. Now because we know that your meiosis 1 had started with a diploid cell, the result was a haploid cell. So this meiosis 2, it can also be called as the mitosis of haploid cells. So this is some information, extra information I am giving you all from the exam point of view. It is also called as the mitosis of the haploid cells. Okay. Now cells that enter the meiosis 2 are the ones they who had passed the meiosis 1. So if there was some problem in the meiosis 1, the cell will not be promoted to your meiosis 2. That is, If you fail in your previous standard, you will not be allowed to enter, or you will not be allowed, even if you are allowed to enter the next phase, you will not be allowed to answer the exam, right? Same thing. So only those cells which have successfully completed their meiosis 1 with flying colors will be allowed to enter the meiosis 2. Okay, these cells are haploid. Okay, all right. So let's see what happens in the next phases. Okay, so this picture is just a representation that the two haploid cells, they have started the journey of meiosis 2. Okay. So let's see what happens in your prophase 2 of your meiosis 2. Now let's try to compare and correlate it with the prophases of the previous two things that we have studied, that is the mitosis and meiosis 1. Now if you remember that there was disappearance of your nuclear membrane, there was condensation or the formation of compact chromosomes. Same thing is going to happen in your prophase 2. Okay, so it is immediately started after cytokinesis of your meiosis 1. Okay, that short interkinesis, and then there is meiosis 1. But this has to start before your chromosomes are fully elongated. Okay, so it is more like your mitosis. Why it is more like mitosis? Because of these 2 points that we have already studied. So basically, you have to remember one thing and do the copy paste thing, the second thing. Okay, but use your logic. So there is disappearance of your nuclear membrane and there is compaction of your chromosomes. So this picture, it shows the same thing, that broken or the damaged nuclear membrane and the spindle fibers. Okay, they have reappeared because from prophase it has to move on to metaphase. So what happens in metaphase is the alignment of your chromosomes on the metaphase plate. Alright. So chromosomes are again, they are getting aligned on the equator, the same thing which we had studied in your mitosis, metaphase of your mitosis and metaphase of your meiosis 1. Then again, microtubules from the opposite pole, they will start moving towards the kinetochores of the sister chromatids. Fine. Okay. So here it's showing the comparison of your metaphase in mitosis, metaphase in your meiosis, and meiosis 1 and metaphase in your meiosis 2. Okay. So basically there is appearance of these microtubules from your opposite poles getting attached to the kinetochores of your sister chromatids, and there is formation of your metaphase plate at the equator. Fine. So next phase is your anaphase 2. So what had happened in case of your anaphase of your mitosis? There was splitting or the pulling apart of the sister chromatids. Same thing is happening in your anaphase 2. Okay, it begins with simultaneous splitting of the centromere of each chromosome, allowing them to move towards the opposite poles of the cell. Okay. Then next is your telophase. Now this is going to be the end phase of your meiosis. Okay, the last stage. Now here your groups of chromosomes, there will be formation of nuclear envelope around this groups of chromosomes. Okay, and finally there will be something called as a cytokinesis, that is separation of your cytoplasm, which will give rise to 4 different haploid daughter cells, because your meiosis 2 had started with 2 haploid cells which were the product of your meiosis 1. So from meiosis 1 we got 2 haploid daughter cells. Now these haploid daughter cells will again undergo your meiosis 2. Okay, it will again undergo the process of meiosis 2 to give rise to 2 more haploid cells individually. Okay, so together we will have 4 different, haploid cells, haploid daughter cells. Okay, So this is what happens in case of your meiosis 2. Okay. So if we can just compare and correlate your mitosis, meiosis and your meiosis and your finally your meiosis 2, meiosis 1 and meiosis 2, we can see there are much similarities. Okay. So if you see this, okay, so we have taken example of oocyte which we had already discussed in your previous session when we were discussing about meiosis 1. This oocyte is going to enter the prophase, meiosis 1. It will complete your prophase 1, metaphase 1, anaphase 1, telophase 1 followed by cytokinesis giving rise to two haploid cells. Okay. One will form the polar body sorry, which will not go any further divisions, whereas the second one, okay, it will enter the meiosis 2. So after this there will be arrest, okay, cells will undergo arrested in your prophase 1, okay. So during the maturity or maturation, okay, they will undergo these processes, okay. So here we will have the prophase 2, okay, metaphase 2, anaphase 2, telophase 2 giving rise to polar body, and the if there is the secondary oocyte, okay. So if there is any fertilization occurring, it will give rise to the zygote. So this is a process that happens. Okay. So let us see what is the significance of this meiosis. So this is going to be a revision again, that this occurs in your gametic cells because its end product or this main purpose is to form the reproductive cells, that is the egg cell and the egg cell and the sperm cell. So this is a reduction division because there is a convergence from your diploid phase to your haploid phase, that is 2n to n. So the chromosome number is getting reduced by half. So it is a reduction division. So in your meiosis, there is an increase of genetic variability in the population from one generation to another. Because see, one generation, okay, suppose there is one generation of the population, okay, they will come from set of two different parents. They will have their own genetic material, the zygote, okay, zygote will have their own unique genetic material again, okay. They will contribute to their own cells, okay, own gametic cells, okay. Then there will be another parent that will come into the picture, okay. So half of the things will come from this parent and half the things will come from this parent. So at each level there will be new genetic variability that will get introduced into the population, okay. So that is why meiosis is so important. Okay, if you see this slide, okay, there is a comparative, okay, pictorial comparison of your mitosis, meiosis 1 and meiosis 2. So if you see the phases are same, okay, if you see all the phases are same. So the major difference will occur is in your meiosis 1 because of your prophase 1. Your prophase 1, there are 5 different sub processes that are sub phases that are happening, and major contribution or the difference is coming because of this prophase 1, because there is crossing over, okay. And from a diploid phase we will have the haploid cells which will enter the meiosis 2 giving rise to 4 haploid daughter cells, okay. And this chart, okay, if you can understand this chart, okay, it is nothing but it is a comparison of each phase of your mitosis, meiosis 1 and meiosis 2. Now if you can prepare this chart, so understand all the three concepts, put it up in one piece of paper, okay, either you can type or you can just write it down so that you will remember what happens at each phase, okay. And also the second second plus point of this chart is that you will have a ready-made comparison of meiosis and mitosis as well as meiosis 1 and meiosis 2. So that in one chart you will have your entire mitosis, meiosis, that is both your meiosis 1 and meiosis 2. You can remember it or memorize it in one page rather than turning the entire pages of your entire chapter. Make your work more simple, more easier by putting up in a comparative table, so that during your exams you don't have to go through the number of pages. Just remember, open this chart, go through it easily. It will be very easy for you to understand and remember it. So till then, keep studying and keep smiling. Thank you.